Our method helps answer the question of how likely a given soil or sediment is to release soluble phosphorus under prolonged saturation. Quantifying phosphorus release potential in variably saturated environments is important for determining transport risk to streams and designing mitigation practices. The main advantage of the method is its ability to simulate important biogeochemical processes affecting soluble phosphorus release and mobility under field conditions.
A scientist trying this method for the first time should not struggle, as its simplicity is one of its main advantages.
To begin, collect approximately 4 liters of soil from desired sites. Limit collection areas to approximately 10 square meters to reduce the amount of spatial variability represented by the sample. Sieve samples through a coarse 20mm screen, followed by a 2mm screen. Thoroughly hand-mix the samples after sieving. Weigh out 100 grams of the field-moist soil. Dry in an oven at 105 degrees Celsius for 24 hours. Weigh the dry soil and calculate percent gravimetric water content. Then, measure out a 500 milliliter subsample using an empty beaker and reserve for chemical analysis. Use the remaining sieved soil for microcosm studies or store in polypropylene bags at 5 degrees Celsius for later use. Use 1-liter graduated polypropylene or other non-reactive plastic beakers as individual experimental microcosm units. Wash beakers in 10% hydrochloric acid and triple rinse with distilled water.